Genomics, saliva collection kit, whole genome sequencing. Let's see how this goes. Remove the cap from the tube. Spit into the funnel. This isn't going to look pretty. And shake the tube several times to mix the saliva with the solution. And that's it. All I need to find out all about my genetics. It's estimated that our bodies contain 37.2 trillion cells, and within each is a copy of code that makes us unique. Billions of genetic instructions, better known as DNA, that makes up a genome. It's what makes me, me. But what if we could use technology to modify that code? The opportunities, well, scientists say, are endless. We could resurrect extinct species, eradicate disease, tackle climate change. We may even be able to live longer, healthier lives. It seems like science fiction, but rapid advancements in genetic technologies are changing our understanding of life. I've sent my DNA here to find out all about my genome.
This is where we do the first quality control step.
I'm hoping my sample wasn't disqualified.
Oh, it was perfect.
I did it right.
It did it right. Yeah.
Okay. A couple of decades ago, labs like this didn't exist. It was impossible for people to get information about their genome. That all changed in the early 2000s.
We are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind.
The Human Genome Project, as it was called, brought together the best and brightest scientists from around the world. Their mission, to map every gene in the human body.
All of us working on the Human Genome Project realized this was historic. This was something we would only do once as a human species, to read our own instruction book. And we were doing it.
Dr. Francis Collins and his team spent 13 years and $2.7 billion to complete the task.
So, Dr. Collins, please come up to the left.
Okay, so you map the human genome and incredible discoveries are made, innovations. Highlight for me some of the ones that you think are most valuable.
I'm a physician, so I'm going to go straight to the medical parts of this. And I have to start with cancer, because cancer is a disease of the genome. And now all of a sudden, we've learned an enormous number of things about cancer as a result.
So we are entering now the sequencing room.
Since the project, the cost and time of sequencing human DNA has decreased significantly.
Currently, this machine is analyzing 70 genomes.
Now anyone can get their genome sequenced in a matter of hours and pay less than $1,000.
One genome produced 96 gigabytes of data.
The data that's going to come out of this
Yes.
isn't going to make a lot of sense to me.
It won't.
To help me decipher my DNA, I met up with geneticist Ansi Yusudas. Feeling nervous.
No worries, yeah.
So this is the report?
Yeah. So let's
Now, my eyes are really bad, so I can't really... You probably know that already.
You're right.
Terrible eyesight.
Because you have vitamin A deficiency.
I've got vitamin A deficiency.
You have insulin resistance. That means you're susceptible to diabetes.
What can I do about that?
It's, less sugars.
This is a bit of knowledge I think I wish I didn't know.
I didn't see that you have like predisposition to excessive alcohol consumption.
Well, say that again. How do you know that? That's in my genes?
Yeah.
That's fascinating and really quite worrying. The Human Genome Project gave us the tools to read and decipher our genetic code. It was a good start. But what came next changed everything.
Was there a moment where you realised that you'd changed the face of science forever?
Indeed, we understood what could be the potential. What was really mind-blowing was really to see how fast the technology was picked up by the scientists. And this is quite unique, actually.
In 2020, Emmanuelle Charpentier and her colleague Jennifer Doudna won the Nobel Prize in Chemistry for their breakthrough discovery while researching a naturally occurring protein in bacteria called CRISPR-Cas9.
It works like molecular scissors in a way that can cleave the DNA. Another explanation could be like software that allows to rewrite a text. The genome is a kind of library of words and letters. It has a code, and CRISPR-Cas is programmable, and one can use CRISPR-Cas to change the genetic code at will.
The discovery opened up a world of possibilities, giving us the tools to potentially cure genetic diseases and disorders from Alzheimer's to leukemia.
Do you remember what you thought at the time?
I was absolutely blown away. This was the kind of game changer that doesn't come along more than maybe once every decade or maybe more. And if we could figure out how to harness it, this might give hope to those people who suffer from thousands of diseases. All of those rare genetic disorders maybe now would be approachable in a new and powerful way.
The excitement of CRISPR was soon overshadowed with ethical questions.
Two beautiful little Chinese girls.
In 2018, Chinese scientist He Jiankui shocked the world when he announced the birth of twin girls whose DNA he genetically modified to make them resistant to HIV.
Oh, everyone was shocked in the community. The CRISPR-Cas technology is not yet ready to be used directly for modifying the human germline.
Since the experiment, which led to He Jiankui's arrest, there have been no reported CRISPR-related experiments done on human embryos. Still, Emmanuelle believes gene editing will change our lives for the better.
There are already a lot of developments of the CRISPR-Cas technology, and this will be developed further. CRISPR-Cas has a very big role to play. It has already played a role, and it will play a very big role in the 10 years to come.
This isn't your ordinary camel farm. Most here are destined to become racing champions or beauty pageant winners.
Rewind. Beauty pageant for camels.
Yes.
What makes a camel beautiful?
Their looks, their lips,
Their eyelashes?
their eyelashes, everything.
Always been jealous of camels' eyelashes.
These camels aren't only being trained to win, they were born to win with the help of genetic engineering.
She's one of the clone babies born last year.
Do you think she knows she's a clone?
I don't know. Maybe.
Never asked her.
I never tried to. Hi. How are you doing today?
Dr. Nisar Wani became the first scientist in the world to clone a camel back in 2009. Fast forward to today, and he says he successfully cloned hundreds.
Baby camels.
These two are the same.
Identical.
They're copies of the same animal. They always will sit down together, they will play together.
While the two are cloned from the same DNA, they were born to two different mothers.
The surrogate mother has absolutely no idea that this is a completely genetically different camel. But when you're here, you kind of forget about all that. What's more natural than this?
Yes.
Thank you. Now what happens in here?
Inside Dr. Wani's high-tech lab, I'm shown the magic, or the science, behind the curtain.
I know now where the DNA is. I went exactly the same place to remove this DNA.
Oh my goodness, you can see the DNA
So
being
see,
sucked up.
sucked out. Done. And inject this DNA into this one.
And that's that. You've just switched the DNA of that egg.
Exactly.
Dr. Wani made the process look so easy, I thought I'd have a go. Not on a real egg, of course.
I'm not good at video games,
No.
so this might not go well.
Yes, go in. Yep. He did it wrong direction.
Oh no. Can I fix it?
No.
What's that saying about you can't make an omelette without breaking some eggs?
Aside from cloning camels, Dr. Wani is building up a gene bank to preserve other animals and endangered species.
There are millions and millions of cells in each box, different cells from different animals.
Should we ever have an animal go extinct?
I don't think so, if as long as we can produce the babies.
Right, you better put the lid back on. Don't want it getting warm.
Not only are we able to save endangered animals, one company believes we may even be able to bring extinct species back to life.
What's really, really interesting is a lot of the technologies that we use today and we're starting to use and evolve in the lab, like CRISPR and other gene editing tools, are directly applicable to de-extinction and to conservation. Conservation works, it just doesn't work at the speed and scale humanity destroys the planet. It just doesn't. It's forecasted that we could lose up to 50% of all biodiversity between now and 2050.
To help solve this problem, Ben Lamb and his company Colossal Biosciences are turning their attention to the woolly mammoth. When these beasts roam the earth, they help maintain grasslands by trampling down snow and redistributing nutrients in the soil. Ben says if we could bring them back, it could help decelerate the effects of climate change. One problem though, 10,000 years of damage to the mammoth's DNA.
So we still don't have 100% of all the DNA for woolly mammoth. So we're looking for all the core functional traits, right? And so an Asian elephant is 99.6% a woolly mammoth already. What are the core genes that we need to de-extinct and put into the Asian elephant architecture to make an embryo that actually exudes all of those cold-tolerant physical attributes like the domed cranium, the small ears, the big shaggy coat that we all know and love.
As this advances, will it become easier? And is that concerning? I guess the elephant in the room here really is the ethics of it. Just because you can bring something back, should you?
Yeah, it's a great question. And the answer is no. Just because you can do it doesn't mean you should. You need to be very thoughtful and cautious about any rewilding, whether it's an extinct species, a critically endangered species, or a species that was previously located in that region that no longer exists there. It's much easier to roll back a several thousand pound animal versus something like genetically modified mosquitoes that once they're out in the wild, they're kind of out in the wild.
What sort of population are we talking about here? How many woolly mammoths are you planning on having?
We want tens of thousands of mammoths that we can reintroduce back into the wild.
When can we expect this to happen? When can I see the first woolly mammoth?
Yeah, so our goal is 2027. We feel really good about that timeline. Like, we've already done the genetic sequencing. It's really exciting.
Genetic technologies are ushering in a new era, from breakthroughs like CRISPR that can edit our genome, to cloning and conserving endangered species. Advancements in the field are even being used to address one of humanity's most pressing challenges, world hunger. Dr. Tarifa Al-Zabi says the fix to solving the crisis could lie in genomic research.
Takes us a year or takes 6 months sometime where we can test different crops.
She's the Director General at the International Center for Biosaline Agriculture, an organization finding ways to grow food in arid environments.
The question is, how can we really feed people with fewer resources, and how will we work with the environment? And so we are quite mindful of the biodiversity, of looking at different solutions that will be environmentally friendly at the same time.
To help find these solutions, the centre recently launched an advanced genomic research lab. Their goal? To sequence the genomes of crops and identify traits that can lead to healthier, more nutritious and resilient produce.
So we have the quinoa here.
Yes, we have the quinoa here, and there are so many blocks with different varieties, so we can test their performance under the different conditions.
The biggest threat to farmers is the worsening climate crisis. One lab in California believes gene editing can slow down its effects.
This is one of our indoor grow rooms. So these are some of our photosynthesis enhanced trees.
Maddie Hall and her company Living Carbon are gene editing trees to help fight against climate change.
You're biohacking trees to do what?
We are genetically engineering trees to capture and store more carbon. Our first product is a photosynthesis enhanced tree that grows faster, captures more carbon, and is more resilient to drought conditions.
Maddie says their photosynthesis enhanced trees grow 39 to 53% faster than normal trees, thus sucking more carbon dioxide out of our atmosphere. This technology also raises concerns about the ethics of manipulating nature at such a large scale.
There's a lot of criticism that people are playing God, mucking around with natural selection, mucking around with nature.
Frankly, we've been mucking around with nature for a really long time as humans. That's what's gotten us into this mess. Just a problem we've created. And even though we've seen large-scale warmings and coolings across geological time, the rate in which this is happening is much faster than ever before.
Maddie believes her biohack trees may be one of the more natural ways to combat the climate crisis.
There's a potential that maybe over millennia, trees could continue to evolve to be more efficient. But what we know about climate change is that we need to address this in decades and not in millennia. So we can't really wait for evolution to have trees evolve to be more resilient to climate change.
This clinic claims to reverse the biological effects of aging.
Oh wow, it looks like a plane.
Yes, so we've got this oxygen mask here.
Okay, just goes over here?
Yes. And for the next 2 hours, you're going to be doing some cognitive training.
By inhaling 100% pure oxygen and taking a series of tests in this pressurized chamber,
I did not do well. I only got 3 stars.
Doctors here say I can lengthen my telomeres, the protective end caps on DNA strands associated with the aging process.
Yes! 4 stars.
Very
Frustrating, those tests. I think I need to go lie down.
Yes.
Our obsession with living forever is nothing new. Civilizations have long dreamt about immortality, from the Fountain of Youth in ancient Greece to tales of the Holy Grail.
Eternal life.
But now people are turning to science to unlock the secrets of aging. Most scientists agree that the main driver of aging comes from mutations or changes to our genetic code. But a new study out of Harvard says that may not be the whole story.
These are human brains grown from stem cells, either from normal or Alzheimer's patients.
David Sinclair is a geneticist at Harvard Medical School and the author of Lifespan.
When we talk about aging, we think of it generally as something very natural that happens to everyone. Is that not the case?
Aging, in my view, is a medical condition that should be treated. The way we look at old age is we slap band-aids on these diseases that aging causes. But what we're showing in this work, heart disease and cancer and Alzheimer's and diabetes and frailty and all the things that we think are typical of old age, we think that there's a universal process that drives all of those. So this is reversal of aging up here and down here.
David's team just finished up a 13-year study that shows that one of the root causes of aging derive from our epigenome, a mechanism in our body that tells our cells how to behave.
As we get older, what we're finding is that nerve cells forget how to be nerve cells and start to look more like skin cells, and vice versa. And of course we're going to get dementia and Alzheimer's disease if our brain forgets how to behave.
Using this data, David says his lab were able to change the biological age of mice.
We found a way to accelerate this aging process by cutting the chromosomes. They had diabetes, they had heart disease, they had memory loss, they had gray hair and wrinkles, and they died a little bit earlier as well. They didn't just look old, we could actually measure the clock, and they were 50% older than their brothers and sisters that were untreated.
And then, even more remarkable, you were able to reverse some of the aging process in a mouse.
I didn't know in my career that we would be able to do this. I thought we'd just be able to slow down aging, and that was good enough. It turns out if you turn on genes that are normally only turned on in embryos, and if we put them into adult animals, we can actually reverse their age in a matter of just a couple of months. We don't fully understand how it works yet, but what I can tell you for sure is that in an old mouse, and probably in an old person, the information to be young again still exists. We just need to know how to flip the switch and reinstall that software to be young again.
While all this information sounds exciting, David is quick to caveat it. Rejuvenating cells in mice is one thing, but will it work in humans? You have famously said that the first person to live to 150 years old is already alive now. Do you maintain that?
No, I don't believe that anymore. I think it's actually older than that. I think we'll learn during the rest of this century how to reverse aging, and probably reverse it multiple times. Now, I'm not saying it's definitely going to happen, but I do know that children born today are going to live into the 22nd century, and we cannot even imagine what that's going to be like.
Rapid advancements in genomics are revolutionizing our world and deepening our understanding of life. By further developing these technologies, scientists around the world think we can save species.
We're starting to bridge the gap between technologists and conservationists.
Fight against climate change.
I think it's actually irresponsible to not use all these tools at our disposal that could potentially help our ecosystems.
And perhaps, more importantly, cure diseases.
You can't help but be excited. This is a therapeutic revolution.
I think I've made a friend. Number 1115. Oh, sometimes we all need a hug.